there. All right, we're recording. We're up and running, folks. Okay. So keep in mind, osmosis, when you see the word osmosis, what do we think? What word do we think? Water. Water. Okay? So, and then it's also a passive process, so it doesn't require energy, and it involves moving down the concentration gradient. And since it's water, water is going to move down its concentration gradient, all right? But in this case, through what we call a semi-permeable membrane, or a selectively permeable membrane, in which what that means is the only thing that can cross over that membrane, or that barrier, is water. So for our purposes, the plasma membrane is going to play that role. Okay, so basically what we're talking about here is, how does water move back and forth across the plasma membrane? Okay, and so you remember me talking about solutes, okay? Those are going to be anything, for our purposes, that are not water. So I like to use glucose, for example. Glucose is wonderful. Okay, we can even use salt if we want to. But glucose, water likes to follow glucose, and I think I talked to you guys about that on Tuesday, how the kidneys, if you're peeing glucose out, water is going to follow it, and that's why people with diabetes are peeing all the time. All right? So when I was talking to you about the concentration gradient, okay, we're going to see the greater concentration of solutes is going to contain the lower concentration of water. And when I was talking about this on Tuesday, and Anna, I know you're at home and you were concerned about this. This is before the movement of water. Okay, so this is before water has even started to move from one area to the other. Okay, so keep that in mind, right? Because again, like with anything in nature, we always want to have balance. Okay, so when we get the movement of water, it's going to move down its concentration gradient. So it's going to move from an area where there's a lot of water to an area where there's less water. But this statement here reveals the relationship between water and solute concentration. Where you have a low concentration of water, you're going to have a high or greater concentration of solutes. So another way of looking at this would be, water is going to move into the area where there's a greater concentration of solutes. All right, and we'll use glucose, for example. Where there's a lot of glucose, water will be drawn to that. Okay, so that's what we're talking about here. So when we refer to the movement of water by osmosis, all right, and how it's dependent upon, all right, that concentration gradient between the cytosol, which is the inside of the cell, and the solution, whatever that cell is in. Okay, if it's in blood, all right, if it's in the body, it's going to be whatever the tissue is going to have. Okay, so the same principle with osmosis, the same principle that we saw with diffusion is going to be with osmosis. Water is going to continue to move until it's reached equilibrium. Okay, it wants that 50-50 relationship. All right, so we're going to talk about how when we have a solution that has 1% solutes, all right, which is a relatively low concentration of solutes, it'll have a higher water concentration versus 3%, that's going to have a higher concentration of solutes, therefore it'll have a lower water concentration. Okay, and that goes back to this. So I would say memorize this statement here: the greater the concentration of solutes contains the lower concentration of water. And that's before water even starts to move. Okay? Once we've established that concentration gradient, it's game on. Water goes from an area of high concentration to low concentration. All right? And that happens to be that low concentration of water, I'll repeat myself one more time, is going to have that greater concentration of solutes. So for this example here, we got 1% solutes, all right, on this side of our membrane, and then we got 3% on the other side. Okay? So this is for solutes. I'll put SOL. So which side, you can say left or right, all right, which side has, are you all in the regular classroom? Yes, we are, regular classroom. Which side, is it the left side or the right side, has the greater concentration of water? The left side. Because remember, the greater concentration of water is going to be where you have the smaller Are you in the right building? Try it again. We're on the 3rd floor, room 344. Oh, you're at home now. Are you at home? Yes. All right. Well, what time did you come by? Were you early? It's all right. How are you doing this? Are you sitting downstairs? Come on up. Join us. All right. Okay. Don't worry. I'll only mark you 10 minutes late. You're fine. All right. Just kidding. All right. So where there is smaller concentration of solutes, we'll see a greater concentration of water. So this has a high concentration of solutes. So will it have a higher concentration or lower concentration of water? Lower. Right. Okay. Okay. Good. And we'll go over some examples here. All right. So let me blow this picture up a little bit so we can see it a little bit better. So you can see here. All right. Here's our plasma membrane. All right. You get it now. Good. Good. These are tough concepts. So sometimes I have to repeat myself 10 times, which is not a problem because it's hard to understand it at first if you've never seen this stuff before. So if you look here, these little red dots, those are the water molecules. Can we all agree that there's more red molecules on the right side of our screen than there are on the left side of the screen? Okay. So where does water want to go? This is the outside of the cell, and this is the inside of the cell. It wants to go inside, right? So here's the thing. Water can actually diffuse, I shouldn't say diffuse, excuse me, osmose or move across the plasma membrane, okay? But it can also go through these aquaporins, okay? How nice is that? When we stick some of these holes, they're pretty much punching holes into the plasma membrane that are made specifically for water, nothing else can go through, then water can start pouring through, okay? So we're seeing the process of osmosis is moving across, all right? Now it's tough to tell. Let me show you, well, I'll show you that picture in a second here, all right? So keep that in mind. Water is going to go from an area of high concentration to low concentration, okay? But if there were more solutes here on the inside, you can also say it's good, and less on the outside, you can say it goes from an area of low solute concentration to high solute concentration. It's drawn, think of the solutes as something that's attracting the water, okay? It's pulling it towards it, okay? So that brings us to osmotic pressure. Very important concept, okay? Because especially this bold-faced word down here at the bottom, hydrostatic pressure, you will need to know that quite extensively in 211, all right? But osmotic pressure, okay, same type of thing, all right, it deals with water, all right? So it has to do with the movement of water across that semi-permeable membrane, but it exerts and it creates a pressure, okay? So this pressure is created, and it's due because of that differential, the difference in concentration, all right, of the solution, okay? So it's the same thing as diffusion in regards to the steeper that concentration gradient, all right, the more water will, the larger the amount of water will move, or the more water will move, all right, and it will create more of that pressure. It's like if you turn the water faucet on a little bit versus a lot. If you turn the water faucet on a lot, water's going to pour out more. It's going to have more pressure. Okay, same thing when we're discussing osmotic pressure. Okay, so if we have a very steep concentration gradient versus this real gradual one, all right, the pressure here will be lower compared to the pressure in this one here. This will have a higher pressure because the concentration gradient is steeper. Okay, and because of that steepness of the gradient, meaning the distance, all right, will generate a greater osmotic pressure. So in our blood, okay, in our vascular system, all right, when you have blood flowing through your blood vessels, your veins, your arteries, your capillaries, it has this term called hydrostatic pressure. And this is the pressure, I apologize for the crudeness of my drawing, all right. All right. When the blood is flowing down that blood vessel, okay, as it's flowing down, it's going to push on the vessel wall, okay? And that pressure that it's pushing on the vessel wall, all right, is called the hydrostatic pressure, It's important to know because when we get to capillaries, capillaries are one cell layer thick. They're made up of endothelium, which is simple squamous epithelium. Okay, so it's one, like in the alveoli, all right? It's one single cell layer thick. So a lot of times, all right, especially in tissues, where how do you think blood gets into the tissues to nourish? You know, how does oxygen get into the tissues, and how does carbon dioxide move from the tissues into the blood? It moves across the vessel wall here, all right? And you want it to be as thin as possible. But the point is, all right, things will move out of the blood vessels because of this hydrostatic pressure. You'll learn more about it. But you want to know what hydrostatic pressure is, and it's that pressure that the fluid in our blood vessels exerts on the inside of the wall as it's pushing outward, okay? All right, so let me show you here, for example, of osmotic pressure, this figure here, 4.12. All right, so here we see this is before anything happens, okay? Side A, side B. We got a nice horseshoe model here, okay? So again, the little red dots, all right, those are the water molecules. The black dots are our solutes, all right? Let's count up real quick on side B. 1, 2, 3, 4 solutes on side B. 1, 2, 3, 4, 5, 6, 7, 8 solutes on side A. We all agree on that? All right, side A has 8, side B has 4. Which side has more? Side A. Okay, so knowing what we know about what water likes to do, where do you think water is going to go? So it's going to flow. It's going to flow this way. Agreed? We all agree on that. Can you guys at home see that? You agree with me at home? Okay. Okay. So when water is moving, it's going to exert a pressure. All right. That's the osmotic pressure from the movement of the water. It's going to flow, and I'm going to show you here, all right, what it's going to look like. So we should see the water level rise, right? Agreed? Yeah. So that's what we're going to see. Boom. Okay. As water pushed its way through, all right, it exerted a force, and that's our osmotic pressure. Okay. And so you can say, we go back to this one here, and if you want to count up all the water molecules on this side and count them all up on this side, all right, you'll see, and you can glance, there's more water here than there is here. Do you agree with me on that at first glance, kind of? I mean, you can go ahead and count it, but just trust me, there's more on the right than there is the left, okay? So again, now we're following the process of osmosis. We're seeing there's a higher concentration of water on side B, and there's a lesser concentration of water on side A, so it's going to flow down its concentration gradient, and that's what we see here. You guys feel a little bit better about that, or am I keeping it too simple and wasting? Should I move on? All right, and the only reason why I keep talking about this is because you're going to see this again in 211. And so I don't want you to sit there and be like, oh man, I don't remember any of this stuff, or I didn't understand that. I want you to understand it now so you don't have to learn it later on, okay? All right. Okay, so that brings me to my next concept here, all right? And that's the concept. We already understand osmosis, but it's this guy here, tonicity. And you're going to want to make sure that you write this down, okay? Tonicity is the ability of a solution to change the volume or the pressure, which we refer to as the tone, so then hence that's why we call it tonicity, of a cell by osmosis. So what that means is, all right, water is going to flow from one area to the other area, okay, and it's going to affect the area that it flowed into one way or the other, okay, and it will also affect the area that it moved away from, okay. So as, for example, if water goes into a cell, the cell is going to swell up, okay, because it's increased the pressure inside the cell. Now, if water leaves a cell, that cell will kind of shrink down, and the pressure has become less inside the cell. So that's what we're talking about with tonicity here, all right? It's the ability of the solution, because we put that cell, and I'll show you here when we talk about the different types of tonicity. All right, we're going to take a cell. We're going to take it for our experiment. We're going to take a red blood cell, and we're going to put it in 3 different types of solutions. Okay, 3 different types, and we're going to see what the solution effects have on that cell. Okay, so what that means is, all right, coming back up here to this bullet point here, So a cell can either gain or lose water through the process of osmosis, all right, depending on whatever solution it's in, and that will change how big a cell is, the volume of it, and also the pressure inside or outside the cell. So let me show you an example. So there's three types when we talk about the concentration of solutes here. There's isotonic solutions, hypotonic solutions, and hypertonic solutions. And for those of you that have worked in a hospital, all right, this will play an effect, because if you've ever had to hang a bag of saline on somebody and you never knew what the heck you were hanging it on them for, you're going to see here for a moment. All right, the first one is isotonic. Iso means the same. It does not change. Okay? So that means the cell that we're going to use in our example is going to be a red blood cell. Okay? So the red blood cell has cytosol. That's the inside of the cell. And then we drop it into a beaker, all right, a solution, all right, that has the same number of solutes that are inside the cell. The same number of solutes will be in the solution. So they'll be the exact same. Okay? So if we have equal concentrations of solutes inside the cell and outside the cell, what's water going to want to do?
Nothing.
Nothing. That's right. Because remember, think about it. It's going to want to flow to where there's more solutes. But if they have the same number of solutes inside the cell versus outside the cell, it's not going to do anything. Okay? So it's not going to go anywhere. So like the slide says here, there's no net movement of water. Okay? So that means there's going to be no effect on our red blood cells, the erythrocytes. And that's what normal saline is. Okay? It has a slight small amount of salt in it, but that's why normal saline is perfect for somebody that is bleeding out in an emergency. They're starting to lose blood volume, but we don't want to affect any of the other tissues in the body. So we just give them normal saline to replace that blood volume. Traditionally, at least males, the normal volume of blood in the body at any one time is about 5 liters. In women, it's a little bit less. You know, I think it's 4.5. Okay? So if you lose a liter of blood, you need to replace that quick, because it takes a long time for your body to replace those blood cells on their own. Because if you don't replace the blood, your blood pressure drops, then you can start to have problems. You can have heart problems, you can have brain problems, okay? You could die, essentially you could die. So we need to replace that blood volume, and we just hang a bang of normal saline on there to replace that blood volume there, okay? And it won't have any effect on any of the other tissues in the body. All right, so the next type of solution is hypotonic, okay? And that means hypo is less. So that means that the solution, all right, the beaker of whatever we put the red blood cell in has a lower concentration of solutes, okay? A lower concentration of solutes, okay? Which means if you have a lower concentration of solutes, then you have a higher concentration of water, right? Okay, remember, there's that relationship there. So what's going to happen, okay? You see outside the cell, I'll just draw a circle here, all right? You have 1, and then inside the cell you have 10, okay? That's for the solutes. Put S. So that means we'll use the same numbers. You have 10 molecules of water outside the cell, and 1 molecule of water, we're just going to put an H, inside the cell. So where is water going to go? It's going to go into the cell. Correct. So it's going to move down its concentration gradient, and so when it moves into the cell, it's going to make the cell bigger, and it's going to increase the pressure on the walls of the inside of the cell. So what's going to happen in the cell?
It's going to expand.
It's going to pop. Yeah, it'll expand and then pop. And that process is called lysis. Okay, the red blood cells will literally burst because they can only handle so much pressure and so much volume. Okay, so when that happens to red blood cells, we call that hemolysis. Yes, ma'am.
So is that where the term hemolytic anemia comes from?
Correct. Hemolytic anemia, obviously, where we're going to have, you're anemic because your blood volume is decreased. Okay, that's the term anemia. And then there's specific types. There's pernicious anemia, iron deficiency anemia, which is IDA. Most people are familiar with that. But hemolytic is literally when the red blood cells are just breaking down, they're popping. okay, for whatever reason, okay? All right, so the last type of solution is a hypertonic solution, okay? Hypertonic, which means it has an increased tone, all right? This means that our solution has a higher concentration of solids, all right? So we'll switch the numbers, 10S versus 1S. 1H2O versus 10H2O. So, where is water going to go? Is it going to leave the cell or enter the cell? It's going to leave the cell because there's less water outside the cell. Okay? So when it does that, it's going to move down its concentration gradient again, and it's going to move from inside the cell to outside, thereby decreasing the volume and the pressure, and it shrinks it. And that term is called crenation. Okay? Crenation. Be familiar with that, all right? The tones, the tonicity, all right? Isotonic versus hypertonic versus hypotonic, all right? And again, sometimes if it helps you to draw it out, then draw it out. That's usually how I learned how to do it. But if you can understand that relationship, that the greater the concentration of solutes, the lesser the concentration of water, okay? You know that, all right? That relationship between that principle there, then you can figure out how water is going to move when you're dealing with a cell and tonicity, okay? All right, so this here is just a picture, all right, of all those examples. So when we have our isotonic solution, all right, you have the same amount of water, solids outside of the cell versus inside. So water doesn't move, so it's fine. And then we have a hypotonic solution, all right? So we have a decreased amount of solids outside the cell. There's more inside, so that's going to draw water in. And the cell will expand and burst. And then our hypertonic has a greater concentration of solids outside the cell, okay? And then what we'll see in that situation is, all right, water will move outside the cell and shrink the cell up. That's called crenation, okay? Go ahead.
Is hypotonic like when someone's in fluid overload or one heart failure? Or
I mean, now we're getting into body compartment physiology, but part of that can occur. Again, when are you talking about like congestive heart failure kind of thing? And again, you have to understand your body's always going to try to compensate. So initially, you can start to get that, but then your body will start to compensate for that, and depending on what the consistency of the fluid. And usually, when you get that edema, like the swelling and whatnot, now we're talking about the oncotic pressure, which is involving proteins, and proteins have an attractive force on water. So then you're starting to get into a couple different things. But it's very similar, but not exactly. I don't want to give you the wrong impression, but it's very similar. What occurs to the tonicity of a cell when placed in a hypertonic solution? All right, the water is going to leave the cell. That's the third example that we saw. There's more solids outside the cell, so water is going to move from inside the cell to the outside. Okay? All right. Whew, that was passive processes. We have far more active processes. But the point being is, it's the passive processes you really don't want to have a good understanding. The active processes aren't going to be. I don't have to go into as much detail. Trust me, it's a lot. Okay, so let's talk about the active processes. Remember the first thing I said about an active process? It requires energy, and it needs it because it's got to roll that boulder up the hill, or it's got to move something up or against the concentration gradient, okay? So, and with active transport, a lot of the times we are creating or maintaining that concentration gradient between the inside of the cell and the outside of the cell, which is the interstitial fluid, okay? Keep in mind, all right, another name for the fluid outside of the cell is called interstitial fluid, and that's the tissue space, okay? All right, so, and we'll talk about, that's not terribly important, but the source of the energy that you have is going to determine whether it's primary or secondary, all right? I wouldn't worry about that too much, but I want to get into, all right, this is one of my favorite things in the human body, All right, I have a lot of favorite things. This is one of them. All right, the ion pumps. These guys are huge. Now you have several. You have ion pumps in your erythrocytes and in your neurons. All right, and you have them in other cells too. But basically, the example that we're showing you here, we have an erythrocyte here that's got a specific ion pump. It's a calcium ion pump. And what it does is it helps to pump, all right, calcium out of the cell, okay? Because one of the things, if you ever take a pathology course, calcium likes to invade tissues, and guess what? Have you ever heard the term calcification? Yeah, okay. Calcification is the process of calcium making something hard and stiff, okay? And when it makes it stiff, all right, it loses, all right, its flexibility. And red blood cells need to be very flexible because they got to squeeze into really small spaces. So if their plasma membrane becomes stiff, they can easily rupture and die, okay? So, all right, your red blood cells were clever enough to put these calcium ion pumps in the plasma membrane, and they just keep pumping that calcium out, okay? But unfortunately, all right, it has to pump that calcium against its concentration gradient, okay? And that requires energy, okay? So keep that in mind, all right? But the calcium pumps are an important structure in the processes of the erythrocyte here. The other one that I love, and we're going to get into this one in much more detail, so don't worry if it seems like I barely talked about this, is this guy here. This is the sodium-potassium ion pump, okay? And basically what it is it's found in our neuron membranes, okay? And what it does is, all right, it pumps sodium out of the cell and it pumps potassium into the cell. And that's part of the reason why sodium is in a higher concentration out of the cell, all right, than it is inside the cell. All right, but remember, if there's more sodium outside the cell than inside the cell, what direction does sodium want to go? It wants to move into the cell. All right, but see, the sodium-potassium ion pump has to pump it out of the cell. So it's forcing it against its concentration gradient. So that's why we need this guy, ATP. So what happens? ATP attaches onto the pump. It causes the pump to change shape and allows sodium to move through. And then what happens is that ATP then gets reduced to ADP, okay? And because of that, it releases the energy, but we've got our sodium outside, all right? So what we'll see now, all right, that phosphate group that used to be part of ATP is still stuck here on the pump, okay? But that ATP here is what we use to kind of open up the protein here to release the sodium. And so now what we're left with is potassium stuck on there. No problem, because once potassium leaves, excuse me, I keep saying potassium. Sorry, you guys at home. Phosphate. I saw the P and I just started going into automa. All right, phosphate, okay? So now the phosphate group is sitting here. Well, that's going to come off the pump. And when it does that, all right, then that allows the potassium here to enter back into the cell because the potassium is sitting outside. And it's waiting for sodium to leave. And then it's going to move in and attach to these, all right, slots here, okay? And then when potassium pops off, it closes the pump down so potassium come in. I said potassium again, didn't I? Phosphate. You guys know what I mean. I'm sorry. I didn't go to bed till 3 o'clock in the morning last night. And that just happened. Go ahead.
So when it's moving back, when the phosphate is moving back through, or not back through, going through, does that require the active? Is that active as well? Is doing all it active or?
Wait, I know I need a banana. Wait, are you talking about the potassium or the phosphate? You're talking about the potassium.
Yeah.
I know. I'm messing you up too. All right, so repeat that again. So it's,
Is it so it requires activation to change the shape of the tube pretty much to get the potassium better,
right.
Potassium through for the phosphate?
Yep.
But for the potassium to go through, is that also active transport or active transportation?
Yeah, so here's the thing. Think of it like a rubber band, all right? When you have to pull the rubber band, you're using energy to do that. So that's what's causing, all right, the pump to go from this configuration to this configuration, okay? So now it's in that, now you have it stretched, all right? So you want it to return back to its original shape, so no problem. we're gonna the phosphate that was sitting here, all right, once that pops off, that will then have, like, letting go of the rubber band, it'll go back to its original shape, and that's what happens. Once the phosphate does that, it returns back to its original shape. But just before it does that, the potassium gets inside of it really quick,
Okay.
and then it allows it to go back. So the
Okay.
only part of that, energy requiring part of that, is to go from this configuration here to this configuration here.
Okay.
Right? And that's the only part. All right, we'll go into more detail on this when we get into chapter 12, the nervous system. All right, so this is basically just a quick thing about the sodium-potassium ion pump. All right, important to know that it's going to move both sodium and potassium against their concentration gradient. Okay, and for every ATP that you spend, you're going to move 2 potassium into the cell and 3 sodium out of the cell. I can guarantee you'll see that on a test. All right, whether it's this test or in chapter or your third test or in your final, you'll see a question on that. Okay, so it's a 3-to-2 ratio. 3 sodium leave the cell, 2 sodium enter the cell. Okay, like Thunderdome. 2 men enter, 1 man leaves. All right, in this case, it's 3 sodium leave, 2 potassium enter. Does anyone know Thunderdome? Mad Max me on Thunderdome? I'm dating myself. All right, that's fine. That's okay. All right, so keep that in mind. We're going to hit more on this. Don't worry about it. If you don't, like, oh, I didn't really get that. Trust me, we're going to nail it in chapter 12. Okay, secondary active transport. This is another type of active transport. Okay, so the same rules apply. We're going to move a substance up the concentration gradient. Now keep this in mind. When any type of, when a molecule is in motion, it's in motion because of kinetic energy. Okay? So when it's moving around, kinetic energy is being generated. Okay, but it's also being used to actually move something around. This concept plays a role here because I'm going to read this so you guys can follow me on this. The kinetic energy that we use to move from the movement of one substance down its concentration gradient, all right, is going to allow the movement of a second substance. Okay, so as we move, this is the nice thing about this type of active transport. We got one thing moving down its concentration gradient. As it's moving, there's energy involved. It's generating energy. So as it's moving, it's generating energy. Well, a second substance will take advantage of that energy, and it will move in addition. Now, there's 2 types, okay? The first one is symport. That means both of those substances, when they're moving, are going to move in the same direction. Antiport is opposite. What that means, one substance is going in, the other substance is going out. All right? But that's, well, that one was a primary pump, so that's a primary. This is secondary. What a secondary means is one substance is doing the work. All right? It's going to move down its concentration gradient. Granted, that doesn't require energy, but the sheer movement of that substance is generating energy. And then the second substance is going to take advantage of that energy, and it allows it to move. So that's why it's secondary. When we're talking about the pump, all right, that's a primary. It's when the movement of sodium does not benefit the movement of potassium. Potassium doesn't get energy. It doesn't move because it's getting energy from sodium. It just happens. It just doesn't work like that. All right. So this is. I think I got a picture on this one. I don't. All right. So let me just repeat that. All right. For secondary active transport, we're going to have one substance that's going to move up its concentration gradient. All right. But it starts with one substance moving down its concentration gradient. As it's moving down its concentration gradient, it's generating kinetic energy. All right. That's energy and movement. And then the second substance takes advantage of that generated energy from the first substance. And then it can either move in the same direction or it can move in the opposite direction. If it moves in the same direction, it's symport. If it moves in the opposite direction, it's antiport. But this type of active transport always involves the movement of 2 substances. Okay? One's going to go down its concentration gradient. The other one goes up its concentration gradient. Yeah.
So like if it was doing something in the cell, it would like two things come out of the cell, or it'd be like one thing comes in, one thing comes out, or
Yep.
two things go in.
Exactly.
Okay.
Exactly. Yep. All right. So another type of active transport, and I just made a video for another section of my online class, and I was talking about exocytosis. All right. So this is a form of active transport. And we saw when I was talking about the Golgi apparatus. The Golgi apparatus, remember, it's the warehouse of the cell. It gets proteins from the endoplasmic reticulum, and it packages them up, and it modifies them, and then it's going to send them to either the plasma membrane, or it's going to set to be, and those proteins will be incorporated into the plasma membrane, or it's going to send it to the plasma membrane so it can expel it from the cell. Okay, so that process of when these, they're called secretory vesicles. I'll zoom in a little bit. All right. So that process, when that secretory vesicle, it's going to approach the plasma membrane, and then it starts to combine with the plasma membrane. All right. And as it's combining here with the plasma membrane, you'll see it's going to start to open up here to release, all right, the contents out of the cell and into the interstitial fluid. Okay? And that process, and you want to know this because there's a lot of different processes that you're going to talk about in several of these chapters that involve exocytosis. And that's an energy requiring transport mechanism. Okay?
Would that be primary or secondary?
That's going to be a primary. Yeah. All right. Another process is called endocytosis. So now we saw exocytosis is when the cell is getting rid of stuff. Okay. Now we're going to find how a cell takes stuff up, because that happens all the time. All right. Cell's got to get its nutrients on, so it's going to absorb things in through the cell. All right, through endocytosis. So in this case, it's going to absorb large substances that are outside the cell. Okay. Very similar to how exocytosis did it, but it's going to reverse it. Okay? So a lot of these materials could be used for normal metabolic processes. All right? Or if we need to repair certain things, parts of the plasma membrane. All right? And also, you're going to see for certain functions, cellular functions, which you'll talk more about in later chapters. All right? There's something that's outside the cell, all right, that needs to come inside the cell so we can maybe initiate maybe production of a hormone or whatever. All right, so all that has to do with endocytosis. Okay, I'll give you a second to write some of that stuff down. Let me just run to the not in the bathroom, water fountain real quick. All right, I know that's a lot to take in. Who would have thunk it that the plasma membrane is involved in so much? You know, when I was in high school learning cell biology, we didn't learn all this stuff. We just learned that it's the envelope that holds the guts of the cell in, that's it. But now you get to learn so much more. Okay, so there's three types of endocytosis. Okay, remember when I was talking to you guys about the macrophages? That was like the Pac-Man white blood cell that goes around and gobbles things up. All right. Well, that process is called phagocytosis. All right, which is like what we refer to it as cell eating. Pinocytosis, the nickname for that is cell drinking. And then we've got our receptor-mediated endocytosis. Let's start off with phagocytosis. This is when there's something large outside the cell. Okay, and so the cell, so say there's like a glucose, well, I don't want to say glucose molecule. Say there's a big lipid out there. All right, and so the cell will start to form these pseudopods. Okay, that will extend around that large molecule, and then they'll enclose it into like a bubble or a sac, and then draw it inside. and then it'll fuse with, you guys remember this guy here, the lysosome, all right? That's its main job. A lysosome is just a digestive organelle. It goes around because it has the proteins inside, the enzymes inside are I don't want to say highly acidic, but they're highly caustic. They like to break things down, and that's what it does. All right. So you can see, all right, that a couple of the cells, like the macrophage, loves to do this form, all right, of endocytosis. All right, because essentially it's going around and it's seeing maybe a bacteria or a virus, and it'll go around and gobble it up that way. Okay. So here you can see a perfect example. All right, the cell sees this big, the cell sees this big particle here, and then it starts to extend these pseudopods around it until it completely engulfs it, and then it forms, I'll go back 1 second, and then it forms this bubble or this vesicle here. And then what will happen is it will combine that vesicle with the lysosome. And the lysosome will start to dump its digestive enzymes inside, and it'll start to break that down. Because it could be a big protein, but maybe that cell says, hey, you know what, I need some amino acids. Amino acids are the building blocks for protein, so it'll start to break it down so we can use the amino acids for something else. Okay? This is a very important process, phagocytosis. You'll see a lot of cells will utilize that. You got it. No, you're good. All right, the next one is pinocytosis, cell drinking. Okay? So we talked about when a cell wants large particles like solids, but sometimes it wants fluid. Okay? It might need some fluid, all right? Maybe some oil, some lipid oil, I'm trying to think, some interstitial fluid, whatever. Okay? Because it requires it for a metabolic process. No problem. All right, so what's going to happen, we're not going to see like one big vesicle get created. We're going to see much smaller vesicles that are made in this case. And so a lot of cells will get, because they can't be, they're selective, but sometimes they can't be too selective, so they just have to grab something from a general area here. All right, so it's not going to be a very specific, or what we refer to as non-specific process. They'll take what they can get. They know it's out there somewhere. They just the cell will just create these vesicles here, all right? So you have these small droplets here, all right? It knows it's out there, so it just makes several smaller vesicles, and it pinches them off, and then it just pulls it into the cell, all right? But when we refer to the pinocytosis, all right, I know this says phagocytosis, I should say pinocytosis, that is going to involve more droplets, liquid, okay? Phagocytosis is going to be more solids, larger particles, okay? All right, and then finally, all right, we've got receptor-mediated endocytosis. All right, this is a situation in which you'll have on some of our cells, on the plasma membranes, you'll have these receptors out there, okay? And the receptor, for a perfect example, and you'll learn about this, and I can't remember, I think it's chapter 27, right, in 211, you're going to learn about how on certain cells, you'll have, you guys have heard of HDLs and LDLs and all that, the good fat and the bad fat, and the very bad fat, and the VLDLs, all right? Well, those are just specific types of lipids, okay, depending on their configuration, all right? But lipids float around the blood, okay? But, because when you go and get your blood checked, and they do what they call a lipid panel, what they're doing is they're just getting an assessment of how much, what lipids are floating in your blood. They have no idea what are inside your body tissues, okay? So, in order for us to get these lipids inside your body tissues, we have to rely on our receptor-mediated endocytosis, because this is your body's way of transporting these cholesterols and lipids into the cells. And it does that by having these specialized receptors on the outside of the cell. And then when these lipids, all right, are floating by, they connect to, all right, that specific receptor, and they're kind of like stuck on there like a fly on fly paper. All right, and then what you'll see here, I'll come back to this slide here. You'll see, blow it up a little bit. All right, you'll see how it's stuck onto this receptor here, and then your cell does the same process. It starts to actually engulf all those lipids or whatever, cholesterol, whatever it wants to take in, and then it pinches it off, and then it throws this protein coat around it. And it's called the clathrin coat, okay? And it helps the cell to identify, all right, what's inside here. It actually labels it for you. How nice is that? Very, very convenient, okay? So this is just another way of specialized cells to be able to take something from the external environment, all right, and then pull it in. And in this case, it's just specific to your lipids, all right, or certain cholesterols, okay? All right, that's it for the plasma membrane. Wipe that sweat off your brow, all right? You would think that the nucleus, because it has all the important stuff inside of it, would be the longest portion of this chapter, but it's actually the shortest portion of the chapter, all right? So we're going to quickly talk about the nucleus here, and then I think we'll be done with this chapter. And then hopefully I can start the tissues just for a few minutes. Don't give me that look. I can't tell what your look is. You have a mask on. I can't tell anybody's look. All right, unless your eyes are open really wide and it's like a frightened look. Yeah, I get that one from my son all the time. All right, so the last one here, remember the three structures of a cell. All right, we have the cytoplasm, the plasma membrane, and then the nucleus. Okay, and this is the largest structure, visible structure on a cell. Okay, and like I said before, it's the control center. Okay, now normally you'll have one nucleus per cell. Some cells have many. You guys, name me a cell that has many nuclei. That's multinucleated. You guys remember? That's true. There could be 1 to 2. So that's skeletal muscle. All right. Some cells will have more than one nuclei. All right. Very good. Some cells won't have any. All right. A red blood cell, okay, or an erythrocyte will not have a nucleus here. Okay. So something that's important to know. Well, I won't say it's important. It's interesting to know. The shape of the nucleus mirrors the shape of the cell. Okay. Remember when we were looking at the cuboidal? It was pretty much spherical. Okay. I know that's not the same shape. It's not going to appear as a square, but it's uniform. What page of the book are you on, Dr. Cobb? The page, 120. Thank you very much. Page 120. Hey, we're in building 120. Coincidence? I think not.
We saw it in the right one before.
No, I wasn't making fun of you. I was like, how is she communicating with us? I was like, that's pretty cool. All right. So, and then remember when we talked about the columnar cells? All right, the nucleus was more kind of oval shaped and elongated. All right, so that's what we're talking about when we're talking about these nuclei will mirror the shape of the cell. But one of the important things, and this is truly important, that's where our DNA sits, right here in the nucleus. Okay, that's your genetic blueprint. That is all the information. So when you're making a protein, all right, for whatever, that information is stored in your DNA. Okay, if you need to replicate certain cells, that information is stored in your DNA in every single cell that has a nucleus. So in theory, if we were to destroy your body completely and save one cell, we could technically bring you back and create a whole you just from one cell. In theory, because all your DNA, all your genetic information is inside the cell nucleus there. Okay? So, in the beginning of this chapter, we talked about the nuclear envelope. Very similar to a plasma membrane, all right? The nucleus has a phospholipid bilayer, okay? And its job is similar, all right, in regards to separating the nucleus from the cytoplasm, okay? And so inside the nucleus, it has its own name, and that's called nucleoplasm. But it has these holes in the nucleus so things can leave the nucleus, and we call those nuclear pores, okay? Because ribosomes are made inside the nucleus, and they got to leave, because how do you think they get out into the cytoplasm? Or how do they get out and attach themselves onto onto the endoplasmic reticulum? All right, so they get made inside, but they need a way out. Okay, but also the nucleus needs, all right, certain materials. Like if it wants to make a protein, all right, we got to get amino acids in there. All right, it needs ions. It's going to need water too, because water is a large content, is in high content, I should say, of the nucleoplasm here. So again, that's what we have the nuclear pores for. All right, and then guess what? Inside the nucleus, we have a smaller structure, all right, and that's the nucleolus. And the important thing that you want to know about the nucleolus is that it makes the small and large ribosome subunits. Okay, so you're going to have a small subunit here, then you're going to have a larger one here. Okay, and I won't go through this. If you want to read chapter 3, you can. All right, if you want to get into transcription and translation, that will explain how a protein is made. But it's because of these two small, all right, not small, but these two ribosome subunits here that allows us to generate a protein strand. Okay, not all the cells will have a nucleolus here. Okay. All right. That might be. So here you can see in our picture here. Here's the nucleolus on the electron microscope here. You can see it here on our drawing. And then you got all this kind of gray matter here. That is your genetic material here. All right, your chromatin. All right, and the lighter, I don't know, that's lighter gray. That's going to be your nucleoplasm. And then you can see all the pores here. Here. And on the outside of the nucleus, you have ribosomes. And some of these pores are going to be continuous with the rough endoplasmic reticulum. So you'll get ribosomes that will come right out, or whatever the DNA will come, not the DNA, excuse me. RNA will come right out of the nucleus and go right into the endoplasmic reticulum so you can make proteins. All right. Well, I want to say in chapter 2, I talk a little bit about DNA, all right, but just a couple things that you do want to know about DNA, all right. First of all right, it is going to be made up of nucleotides. Those are going to be the monomers. Remember how glucose, when we started to stack glucose along itself, we got glycogen. Well, the glucose is the monomer. Well, the nucleotides are our monomers for DNA, okay? And it's made up of, and here's the actual ingredients for DNA. We need a sugar, and it's a 5-carbon sugar. All right. You also have a phosphate, and then you'll have 1 of 4 of the nitrogenous bases there. All right. So adenine, cytosine, guanine, and thymine. All right. All 4 of these nitrogenous bases, and they'll vary. All right. And it creates your genetic code. All right. I'm not going to get into the genetics too much on that right now. All right, but what I want you to know is what makes up DNA. All right, you have to have your ribose, your 5-carbon sugar. All right, a phosphate group, and then a nitrogenous base. All right, don't worry about that, and don't worry about that one. Oh. All right. One more slide after this one. Cell division. Okay? Basically on this slide, the only things I want you to know, all right, are there's 2 types of cell division, mitosis and meiosis. And here's an easy way to remember it. Meiosis involves sex cells. Mitosis involves everything else. Simple. Okay? What are the sex cells? Oocytes, the eggs, okay, and sperm. That's it. And then mitosis is all the other cells. And we refer to those other cells as somatic cells. All right? That's the easiest way. Why does cell division occur? 1, during growth. 2, when you want to replace something that's old and damaged. All right? 3, for regeneration. Okay? All right, and last slide. What is the function of the nuclear pores? It's to allow passage of particles into and out of the nucleus. Questions? It's a lot. We made it through though. And I feel, at least from, and I can't see you guys at home, but I really feel like at least you folks pretty much understood the more salient or important concepts for osmosis and diffusion. And that's, trust me, it's going to be very important. I can't stress that enough, especially in later chapters. You're going to really want to understand that. I'm trying to make your job easy. I don't want you to have to struggle when we get to some of the more advanced concepts. All right, let's just kind of quickly go through. We'll start off chapter 5 here with tissues, okay? Because it won't be anything too difficult. And you've seen some of this stuff. So it's going to be almost kind of like a review, okay? So now, as you can see, as we're going through, all right, the organization chart of biological organisms, we started off with the simplest ones, atoms and molecules, then we moved up to macromolecules, then to cells. Now we've graduated onto tissues. So now what we've done is we've grouped some cells together, all right, and they're going to perform a common function, okay? So here's the thing, they're going to be similar, all right? The cells are going to be similar, they're going to have a common function, and then we're also now going to add some what we refer to as extracellular material. You guys remember we were talking about connective tissue? There was 3 components to the connective tissue, all right? Cells was one, all right? And then there's 2 others, but when we combine them together, we referred to them as extracellular material. You guys remember that from lab? If not, it's okay. It just means you weren't listening to me. So the 2 components for extracellular material, remember protein fibers and ground substance? Okay? All right, so we'll get into that more. So know this, the study of tissues is histology. Very important to know. And I cannot repeat this enough times. You need to know from off the top of your head all 4 tissue types in the body. You need to know it. Okay? Epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Just know that those are the 4 tissue types. All right? In this chapter, we're going to go through all the specific ones. Okay? Very important. For those of you at home, I hope you're listening to me. All right. So we're going to start off by talking about the first type of tissue, epithelial tissue. So there's going to be a little bit of review, but I'm going to get into a little bit more of the details, and you'll know most of them. You'll be surprised. Okay? So epithelium, also known as epithelial tissue. Couple of the characteristics, and a lot of these you already know. One, they're closely packed, right? Remember when we saw all the epithelial cells? They were right up against one another, all right? Simple cuboidal, simple columnar, they were all jammed together, all right? There wasn't a lot of space, if any space, in between the cells, all right? So we'll have one layer. What do we call epithelium that's one layer thick? Simple, right on. What do we call it when it's 2 or more? Stratified, yeah, like cloud, stratified cloud. All right. So they're closely packed. They're going to be 1 or 2 or more layers thick. Okay. They will have very little or no extracellular matrix. What that means is they will have very little or no protein fibers. Okay. And what's the other component to the extracellular matrix? Booyah, ground substance. I'm just going to call that GS. Okay? Keep that in mind. It's an easy way to differentiate epithelial tissue from the other tissues. Okay? They're going to be closely packed, and they're not going to have protein fibers or ground substance. So if you see a ton of collagen fibers on the slide, you better not put down simple epithelium. All right? Or stratified squamous epithelium. You know what I mean? Okay? Don't get that stuff wrong. All right, so epithelial tissue is going to cover some body surfaces. Give me one body surface that it covers. I'm looking at it right now.
Skin.
Booyah, skin. Yep, darn right. Okay, the lining of your stomach. All right, it's going to be on inside and outside of body surfaces. All right, it's going to line some of your body cavities. Remember what we learned about. What's the name of the covering of the body cavities? Do you guys remember? All right, I'll be a little more specific. There's 4 membranes in your body. We learned the first one on the first day of lab. It's the name of a membrane. Serous membrane. Remember we learned that there's a visceral layer, and that's the layer that covers the actual organ itself. And then there's the parietal layer, and that's the layer that lines the internal surface of the body cavity. You want to know that, all right? So what we're seeing is that's epithelial tissue, okay? Now we haven't learned it yet, but we're going to talk about glands, all right? So it's going to make up a lot of the glands, and then this is huge, 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 huge. Epithelial tissue is avascular. Remember when we were labeling the skin? All right, we were learning about the epidermis, and the epidermis is all epithelial tissue. You didn't see any blood vessels in there, did you? So, stands to reason. You're flipping through your notebook one day or your book, and dang, you give yourself a paper cut, and it's not bleeding. How far did that cut go down? That's right. It only sliced through the epidermis. If there's no blood, then you didn't make it down into the dermis, okay? Because you don't have blood vessels in your epidermis. There you go. Okay, avascular. So that's important to know that. Okay, so some of the other characteristics here, and I think I did talk to you guys about polarity, but I didn't really talk to you about cellularity, okay? And cellularity has to do with this concept that the cells are packed tightly close to one another, and we have very minimal to no extracellular matrix in between those cells. And so we refer to that as cellularity. All right? Polarity, we already discussed. All right? There's a top part to the cell, which is the apical surface of the cell, and then there's a bottom part to the cell, and that's the basilar surface. All right? And the same thing works if it's stratified layers too. You have the top layer, the apical layer, and then you have the bottom layer, which is the basilar layer, okay, like we saw in the epidermis. The basilar layer to the epidermis is the stratum basale, all right, and then the apical layer was going to be the stratum corneum, okay? So on the apical surface, that's where we're going to see some of those modifications there, or those plasma membrane extensions, like the microville and the cilia, all right? And they're going to be exposed to the external environment, like our skin, how that's exposed to this room's environment. And then when we're talking about the serous membranes or the lining of your intestines and stomach, all right, that's what we're referring to the internal body space there, okay? So when these cells are so closely packed to one another, they get held together. Remember when I said when the keratinocytes, when they're being developed in the stratum basale, and then they migrate into the stratum spinosum, that's the next layer, okay? Well, in that layer, they're starting to become tightly packed to one another, and they're creating what we call intercellular junctions. So they're starting to attach themselves to one another. And that's what we're seeing here. The lateral surfaces of these cells are going to be involved in these intracellular junctions, so nothing can squeeze in between. Because when that happens, you got problems. You're making yourself a victim or a possible victim for infection, okay? And then, of course, you got the basilar surface here, all right? And that's going to be sitting on top of what we call the basement membrane, also attached to the connective tissue. All right. So, a couple more characteristics here. All epithelial tissue, all right? If it's simple, every single cell is going to attach itself to the basement membrane, okay? If it's stratified, only the basilar layer will attach to the basement membrane. What the basement membrane is, it's a selective barrier that's between the epithelium and the underlying connective tissue, okay? Now, I won't ask you this here, but there's 3 layers to it. But just know that it's a selective barrier, which is going to play a role in processes like diffusion, okay, or osmosis, right? It'll allow the passage of certain substances, okay? We've already talked about avascularity. Keep that in mind. It's huge to know that, okay? One thing that you should also know, even though it lacks blood supply, it's avascular, it has a high innervation of nervous tissue, okay? When we were talking about the epithelium in the skin, remember the tactile cells, all right? That was one of the 3 cell types in the stratum basale, all right? So we're going to see that epithelium is going to have lots and lots of nerve tissue there, okay? And then finally, all right, epithelium is getting replaced constantly because it's in places that are undergoing, especially if it's stratified, lots of mechanical stress, all right? So it's going to be regenerating at a high rate, okay? So you'll see its cell turnover occurring quite a bit and quite frequently. Okay, so this next picture here is just showing you here how epithelium, it's showing the polarity here, all right? We got our apical surface, our basilar surface, and then our lateral surfaces here. You can see how they're just tightly packed. Right below that, there's our basement membrane that sits right on top of that connective tissue, okay? No blood vessels in here. All right, a couple functions for epithelial tissue. All right, we've mentioned this in lab. Physical protection. When it lines body spaces, body cavities, and the external surfaces, for example, I'll keep using the skin because it's a great example. All right, when it lines those internal and external surfaces, all right, it's going to offer one, protection from abrasive forces, like when you scrape up against something. All right, if that wasn't present there, you could damage some of the tissues there. But one of the big, huge functions of skin is to actually hold, all right, our water content, or a majority of it, inside of our bodies. Remember what I said? I don't know if I told this group this, but water is not, excuse me, skin is not waterproof, it's water resistant, okay? So it can take in a certain amount of water, but also it holds in a lot of water, all right? Otherwise, all right, if you didn't have the epithelial characteristics that your skin has, you would lose a significant larger amount of moisture from your body than what you already do. You lose a small amount through, all right, we'll talk about it, it's called transpiration, that your sweat glands just give off a little bit of water, just a small, minute amount, very, very small, all right? The majority of water loss occurs through urination. You lose a small amount every time you exhale. That's why we're all wearing these things, all right? That you do, they're more worried about the water droplets, but you do lose some water every time you exhale through water vapor, all right? Some through sweat, and then some through transpiration, and then even some through your feces, all right? But anyways, it helps to prevent you from getting dehydrated, all right? We'll talk about this. You'll see this quite a bit when you get into digestion and in respiration, how epithelial tissue, especially when it is simple epithelium, because it filters, all right, and it also allows for absorption, but it'll, especially, perfect example, the alveoli. When the respiratory gases cross over your alveoli, the air sacs there, all right, that is dependent on the actual construction of that epithelium there. When we get into glands, we'll talk about secretions, and then, like I said, sensations, because the epithelium is highly innervated by nervous tissue, all right, and we refer to that as neuroepithelium, all right, mainly touch, that these types of cells, this type of epithelium, will elicit several sensory information signals to the brain. I think this is a good place to stop here. I wanted to just kind of get through the basic stuff. Does this not look familiar though? Remember we were doing this in lab? So a lot of this is going to look familiar. So don't you guys worry. We're going to move through at not a fast pace, all right? But trust me, you won't be surprised. A lot of this you'll recognize. Okay, so this is almost, next class will almost be a review for you. Okay, let's take a break. All you guys at home, if you have any questions, just hold them until lab.